Good morning everyone. I am Dr. Kalpana Sharma, Associate Professor, Department of Zoology, S.P.C Government College, Ajmer. बच्चों, मेरे कल के लेक्चर में मैंने आप लोगों को या लास्ट लेक्चर में मैंने आप लोगों को सेल साइकिल का भाग पढ़ाया था, जिसमें हमने पार्ट 1 देखा था, जिसमें माइटोटिक डिविजन्स हमने पढ़े थे. आज हम डिस्कस करने जा रहे हैं सेल साइकिल का पार्ट 2, दैट इज मियोसिस. Meiosis is a specialized and rather complicated type of cell division occurring only in the diploid reproductive cell and results in the formation of haploid sex cells or gametes. The process of meiosis. The process of meiosis is separated into sequence of events similar to those of mitosis, but these events or stages are repeated twice, or in meiosis, two complete cell divisions follow in close sequence, which is without a short interface between them. So, the meiotic division, it is divided into two types. One is heterotypic division or the reduction division, and the second one is the homotypic divisions or the equational divisions. Heterotypic division or the reduction divisions are divided into prophase 1, metaphase 1, anaphase 1 and telophase 1. The prophase 1 of the heterotypic division is divided into further divided into 5 stages, that is lactotine, zygotine, pachytine, diplotine and dikinesis. Whereas the homotypic divisions or the equatorial divisions, it is divided into again the 4 phase: prophase 2nd, metaphase 2nd, anaphase 2nd and telophase 2nd. In this picture, you can see the diagrammatic representation of different stages in first meiotic division. So first of all, we are going to discuss the first meiotic division, that is heterotopic divisions or the reduction division. In it, the diploid parent cell divides into 2 daughter cells having haploid chromosome number, the prophase of which is divided into 5 phases: liptotine, zygotine, pachytine and diplotine. Now first of all, we are going to see the heterotypic division. The first stage is the prophase 1. The prophase is quite lengthy because it contains the 5 another stages also. During prophase 1, the homologous chromosome pair and exchange DNA to form recombinant chromosome. Prophase 1 is divided into 5 phases, that is liptotine. Liptotine here, then this you can see the diagram of the liptotine. Chromosome starts to condense. So during the liptotine stage, what happens that the liptotine stage initiates meiosis. Due to the condensation of chromatin matter, the chromosomes appears in diploid number as long, thin and uncoiled threads of cylinder filament, longitudinally single rather than double as in mitosis. Second stage is the zygotine. Zygotine or the zygonema stage. The zygotine commences with the movement of the chromosome. It is affected by the force of attraction between the 2 homologous of a chromosome pair. Thus, the chromosome of a pair approaches each other and each chromosome shortly takes a position along the side of its partner to form a bivalent. The pairing of homologous chromosome is known as synapsis, and it is very intimate and precise. Third stage is the pachytine stage. Pachytine stage or the pachinema stage. With the pairing or synapsis of homologous, the nucleus enters the pachytine stage. During pachytine stage, crossing over or between the 2 pair of homologous chromosomes takes place to form the chiasmata. Next stage is the diplotine stage or the diplonema stage. The separation of homologous chromosome initiates diplotin, the synaptic force of attraction between them lapse during the breaking at one or more points, so that the homologous chromosome uncoil and start separating. But the separation is nonetheless incomplete, since the homologues are in contact at each one or more points where the crossing over has already taken place. These points of contacts are known as chiasmata, which present cross-shaped appearance, and the chiasma is the morphological equivalent or genetic crossing over. Next one is the dikinesis. In dikinesis, homologous chromosomes continue to separate and chiasmata move to the end of the chromosome. The prophase first is followed by prometaphase 1. During prometaphase 1, the spindle apparatus is formed and chromosomes attaches to the spindle fiber by kinetochores. Then comes the metaphase 1 stage. During metaphase 1 stage, homologous pairs of chromosome or the bivalent, they arranged as a double row along the metaphase plate. The arrangement of the paired chromosomes with respect to the poles of the spindle apparatus is random along the metaphase plate. This is a source of genetic variation through random assortment, as the parent and maternal chromosomes in a homologous pair are similar but not identical. The number of possible arrangement is 2n, where n is the number of chromosomes in a haploid set. Human beings have 23 different chromosomes, so the number of possible combination is 223, which is over 8 millions. Then comes the anaphase 1. During this phase, the homologous chromosomes in each bivalent are separated and move to the opposite poles of the cell. Telophase 1. The chromosomes becomes diffused and the nuclear membrane reforms. This is followed by cytokinesis, or the division of the cytoplasm. The final cellular division to form two new cells, followed by meiosis second. Meiosis first is a reduction division. The original diploid cells had 2 copies of each chromosome. The newly formed haploid cells have 1 copy of each chromosome. Meiosis first is followed by meiosis 2. Meiosis 2 is also known as homotypic divisions, as we already see. Homotypic division is divided into prophase second, metaphase second, anaphase second, telophase second, and cytokinesis cell division occurs. During prophase second phase, as you can see that the chromosome begins to condense, nuclear membrane dissolves, and spindle fiber forms. During metaphase second, the spindle fiber attaches to the chromosomes. Chromomeres line up in the center of the cell. In the third, you can see anaphase second. Centromere divides and sister chromatids move to the opposite end of the cell as spindle fiber shorten. During telophase, as you can see in the diagram, that chromosomes reaches opposite ends. Nuclear membrane reforms. It is followed by cytokinesis, or the division of the cytoplasm, where the cytoplasm divided into 2 daughter cells. Meiosis second separates each chromosome into 2 chromatids. The event of meiosis 2 are analogous to those of mitotic divisions, although the number of chromosomes involved has been halved. Meiosis generates genetic diversity through the exchange of genetic material between homologous chromosomes during meiosis 1, the random alignment of maternal and paternal chromosomes in meiosis 1, and the random alignment of the sister chromatids at meiosis 2. Thank you.